Welcome, and thank you for joining us. My name is Dean Lee, and I'm Professor of Physics at the Facility for Rare Isotope Beams, or FRIB. Today's public talk is part of an initiative at FRIB called the Advanced Studies Gateway. The goal of the Advanced Studies Gateway is to inspire people. We bring together researchers, innovators, creative thinkers, artists, and performers from all fields and strengthen ties between Michigan State University and the community. Today we have the great pleasure of presenting a public lecture by Professor Nigel Goldenfeld, who is a Chancellor's Distinguished Professor of Physics at UC San Diego. Professor Goldenfeld joined UC San Diego in 2021 after 36 years at the University of Illinois at Urbana-Champaign. His research spans condensed matter theory, the theory of living systems, hydrodynamics, and non-equilibrium statistical physics. He received his PhD in theoretical physics from the University of Cambridge and was a postdoctoral fellow at the Institute for Theoretical Physics, UC Santa Barbara, where his work on the dynamics of snowflake growth helped launch the modern theory of pattern formation in nature. He joined the condensed matter theory group at Illinois in 1985, where his work was instrumental to the discovery of D-wave pairing in high-temperature superconductors. In 1996, he co-founded Numerics, a company that develops high-performance software for pricing and risk-managing derivative securities. He was a founding member of the Institute for Genomic Biology at Illinois, where he led the Biocomplexity Group and directed the NASA Astrobiology Institute for Universal Biology. Professor Goldenfeld was awarded the American Physical Society's Leo P. Kadanoff Prize in 2020. He is a Fellow of the American Physical Society, a Fellow of the American Academy of Arts and Sciences, a Fellow of the Royal Society of the United Kingdom, and a member of the U.S. National Academy of Sciences. If you have any questions during this talk, please type them into the chat or question and answer dialog box. At this time, it is my great pleasure to introduce Nigel Goldenfeld.
Thank you very much, Dean, for inviting me. It's a great pleasure to participate in your exciting series. Let me share my screen and let's get started. So why does life exist? The existence of life is the most remarkable and unexpected phenomenon of physics. It's a physics phenomenon. I'm just going to hide this, sorry about that. It's a physics problem because we want to understand why life exists in the first place, not, for example, how to fix a broken kidney in a human that has evolved after billions of years of evolution. So I'm going to tell you today that even though we do not yet have a clear answer to the original question, some puzzling aspects of the early evolution of life on Earth can be understood using approaches rooted in theoretical physics. So I'm going to tell you then about what theoretical physics can tell us about the origin and evolution of early life. And this work was in collaboration with a number of colleagues and students and postdocs over the years at the University of Illinois at Urbana-Champaign, and they are listed here along with the funding agency. So since we're trying to understand what theoretical physics can tell us about the origin and the evolution of life, which you might think is a pretty surprising thing that it can tell you anything about that, let's just ask what can theoretical physics tell us about anything? In fact, what actually is theoretical physics? So let's take a look. So here's a picture, and the question I want you to think about is this theoretical physics? So you probably take a look at this and you say, well, sure, it looks pretty complicated. What the heck is all of that? So maybe it is theoretical physics. It's my blackboard, so I'm a theoretical physicist. So yes, it probably is theoretical physics, but not all of it actually. So for example, this blackboard was photographed when I was still in the Institute for Genomic Biology. So maybe it's not theoretical physics. Who knows? Theoretical physics looks baffling and strange to outsiders, but it's like looking at a game of chess. If you understand the rules, then you can look at the board and immediately see what's going on there. If you don't know anything, it seems very obscure. So let's take a look at this bit of my blackboard. and ask the question, is this theoretical physics? Well, it's not actually. This is mathematical physics. It's not theoretical physics. Okay? It's just really a complicated calculation, like basically a maths homework problem, but just done at the level of PhD or harder than that. Okay, so that's not theoretical physics. That's not so great. How about this bit of the blackboard? Well, this bit actually is theoretical physics. Why? Well, this is a guess about how something behaves in nature. In fact, the thing it is a guess about is in fact to do with how fluids become turbulent, which is one of my interests. So basically what I've just shown you is that, or at least told you anyway, is, and I ask you to believe it, is that this bit of the board is mathematical physics, this bit of the board is theoretical physics. They look perplexing, but they're not the same thing. And what we think of as theoretical physics is basically a simple guess plus mathematics. Okay, that's great. So does it work? So here is the three recipients of the Nobel Prize in 2016. That was awarded in the area of condensed matter theory, which I work in. These three scientists are in some sense Margaret Thatcher's gift to American science. They all won the Nobel Prize. They were from England originally, but they moved to the US in a brain drain inspired by funding cuts in science in the Conservative administration of Margaret Thatcher. You can think to yourself, if you wish, about possible parallels to today's situation. Regardless of that, what did they actually do? Well, what they did was they looked at superfluids. At least that's one way to talk about what they did. So what is a superfluid? Well, on the left here is a fluid that you might have had with your breakfast pancakes today or waffles. Honey, which is a normal fluid with high viscosity at room temperature. It flows very slowly, it's sticky, and you all know it. Here on the right-hand side is a picture that you probably don't know. This is superfluid helium. It has no viscosity, effectively, at 1 degree above absolute zero. And here what you're seeing is a weird effect that happens in superfluid, which I'm not going to go into, called the fountain effect. So here you have a fluid that has no viscosity when you're very close to absolute zero. Okay, now here's a cartoon. This is a cartoon, and it just is a picture of arrows drawn on a square lattice, just like you can see. And the only thing interesting about this picture is that the idea is that if I have two arrows, they could represent, doesn't really matter what they represent. You can think of them as representing birds moving, flying through space or something like this. But the main thing is to know that if the arrows are parallel to each other on adjacent sites, then they have no energy. And if they are slightly deviating from each other, as you can see here, then actually they have a higher energy. So that picture of arrows on a plane turns out that it can predict the aspects of the phase transition between a normal fluid, a regular fluid, and a superfluid, at least if you're in 2 dimensions. I'm not going to go into what is the details shown over here, but this is a graph that basically was the confirmation of the theory of these 2-dimensional superfluids, and the measurements. Some of these measurements on this graph were done by Jacques Michel at the University of Illinois at Urbana-Champaign, and this was a great triumph of thought, and yielded experimental predictions and great understanding, and that's why the people involved in developing this field won the Nobel Prize. So it's pretty strange, because what I've just told you is that superfluids in 2 dimensions, it sounds pretty complicated, very exotic, there's all sorts of quantum mechanical effects and so on, and yet Koster's and Thoules's Nobel Prize winning work that predicted the strange aspects of this phenomenon came down to just looking at the simplest way that arrows on a plane could be described energetically. And the moral of this story is that very simple models, absurdly simple models, can predict very complex phenomena. In fact, everything we know about the physical world comes from looking for the simplest mathematical models that have the right symmetry and topology and various other constraints. And so this idea that simple models can predict complex phenomena is really the foundation of why we can do science. So what can theoretical physics tell us about the evolution of early life? Can simple models of the sort that I just showed you predict real biological phenomena, just like we do in condensed matter theory? Well, the thing about the phenomena in condensed matter theory is that they are in some sense universal. They tell you about aspects of matter that don't really depend on what are the atoms or the molecules in which they are made. So I told you that to understand a superfluid, you just had to look at arrows on a plane. You didn't even have to know that there are atoms or molecules. So it's very strange. There are properties that are what we call universal. They transcend the microscopic aspect of what the matter actually is. So you could say, well, is anything universal in biology? And then you go and talk to a biologist, and the first thing they'll tell you is everything in biology has an exception. So that's true. So you might say this whole enterprise is doomed, and we can finish the talk right now. But there's more to be said about it. So I'm going to say to you a little bit about what universal biology is, and I'm not going to go through all the scientific evidence that's showing you that there are universal phenomena, but I want to say to you what are the aspirations of this field. So let's have a look at a news report that came out maybe 10 years or so ago on the NASA website. And it says this: the mystery of the missing waves on Titan. So maybe you were worried about that, maybe you were losing sleep about it. But what astrophysicists, planetary scientists were worried about was that they know that there's a lake and oceans on the surface of Titan. It's a moon of Saturn, and one thought that there should be waves on that surface, and there's the mystery. They're not there. We can't see water waves like we see the waves on the ocean. But then don't worry, because a little bit later, a few months later, Cassini spies the wind-rippled waves on Titan. So you can sleep tight. In fact, there are waves on the oceans of Titan, and we were able to see them, we as a scientific community. So we know the waves on the ocean of Titan are missing because there is a general theory of two-phase fluid flow interfaces, fluid mechanics, aerodynamics, that predicts these waves and predicts all of their characteristics, and we can then look for them and in fact identify them. So here is a headline that I really would love to see someday. Mystery of the missing life on Titan. Okay, anybody ever seen that headline? Nope. And then how about this one? Even better. Cassini spies long-sought life on Titan. Okay, well, that's not maybe so much science fiction as we'll see as we get into this talk, but why does that seem preposterous? Well, we don't know if there is missing life because we do not yet have a theory that predicts the existence of life as a physical phenomenon, nor do we understand what sort of life can arise in different environments. So that's just a science fiction, it's just a joke, but it's an aspiration. Now, when we talk about universal biology, there's not that much that one can say about it, so I'm going to leave the rest of this slide blank. But there is something we can say about something else which is unexpectedly universal, and that is computation. So let me talk about universal computation. You might think that universal biology might have something to do with universal computation, so let's ask ourselves what universal computation is. So here is my computer, or something like my computer, probably like yours. On the other hand, if you were living in the the end of the 19th century, you might think that a computer was this, a machine that's built out of cogwheels and springs. This was Babbage's computer. You can see it in the Science Museum at London and in the Museum of Computation in Mountain View, California. It was never really completed, but if it had been completed and finished and built, not only would it have been very large, but it also would have been equivalent to everything that your laptop can do. So a computer is neither a shiny chunk of glass, plastic, and silicon, nor is it a bunch of cogwheels, springs, and levers. It's an abstract concept. It's a mathematical device, a Turing machine with a von Neumann architecture that can be instantiated in many ways. And the idea of a computer was first developed by mathematicians like Alan Turing and others, and then was only later built by engineers and, for example, and physicists. And here is a picture in the middle here of von Neumann standing in front of one of the first computers that was built at the Institute for Advanced Study in Princeton to solve mathematical problems to do with atomic weapons. Okay, so the medium is not the message, it's what it actually does, and that can be instantiated in many different ways, and that same thing could also apply to life. Now, Niels Baricelli was a visitor to the Institute of Advanced Study in Princeton during those years, and he built the first computer viruses. His idea was this: if I look at the core memory of these computers, they have memory, they have programs, the programs do operations on the memory. But what happens if the program itself is the data and the data itself is the program? I realize I should be using plural, but most people seem to think that data is a singular word. So I'm just saying the data is the program. In other words, the idea is that the computer program acts just not on data cells stored in memory, but acts on the cells that store the program itself. And what he discovered was that most of the time, the whole thing just crashed and burned. But occasionally, you would get what were, in fact, the first computer viruses that could program themselves and would proliferate throughout the system and obey some of the rules of evolution and ecology. And this is a field that has been really highly developed in the last 20, 30 years by theoretical biologists, including Rich Lenski and Chris Adami at Michigan State University and others too. So this is a very active field of trying to understand the intersection between biology, computation, and the theory of life. So basically, living systems then have the capacity to evolve, and one can think of that as that they can reprogram themselves, just like I was showing you there, and so they're able to respond to environmental perturbations by creating new functionality. And so this is very different from physical systems, which obey fixed laws that are predetermined and can never change their laws. And biological systems have this self-programming capability, and that makes it very difficult to control. Now, in universal biology, we have the inverse problem. The inverse problem is like this. Suppose I gave you a modern computer and I said, could you work backwards and deduce the abstract mathematical theory of computation that Turing and von Neumann and others developed? And that would be obviously a pretty difficult thing to do, because you look at the computer, it looks like a bunch of silicon chips and liquid crystal displays and whatever, and it'd be hard to say that thing is actually instantiating a mathematical process. So in biology, we have the inverse problem. We already have the biology, and we can see it, and we want to understand what is the abstract theory of which it is an instantiation, and that abstract theory would then underlie all systems that exhibit the characteristics of life, both here and elsewhere in the universe. So evolution is special. It's a special characteristic of living systems because complex systems never stop. They keep on evolving. This is called the open-ended growth of complexity. And these are aspects of complex biological systems that those of us who think about these things would love to try to understand. Now, the thing about physicists is that they like to work on simple problems first. Just like Einstein said, your problem should be as simple as possible, or a theory should be as simple as possible, but not simpler. So if we want to try to understand what are the simplest forms of life and how they work, we shouldn't be looking at modern day organisms, we should be looking at early organisms. And so that forces theoretical biologists like me and others to start worrying about questions like how did life get started and what aspects of that we could possibly try to understand dynamically and mechanistically. So I want to now talk about the evolution of the genetic code. I'm going to say what the genetic code is and what it tells us. So here you might say as well, this is obviously the genetic code. It's a bunch of letters, and then you look at them more closely and you see there's A and T and G's and a few C's. And so you might say, well, that's the genetic code. Okay, this is not the genetic code. Okay, the newspapers will tell you it is the genetic code, but it's not. Okay, this is a genome. It may be a fraction segment of a gene. Let's just see what that thing is. So what Francis Crick and James Watson discovered in the 1950s is that there are four molecules that structure DNA. They have names: adenine, thymine, guanine, and cytosine. They're represented by A, G, C, and T, and they form sort of pairs, and this forms the DNA double helix structure that people have heard about. But what is the actual genetic code? What do I mean by that? Well, to understand that, we have to look at what is a process called translation. So if you look at a cell has the blueprints of the cell are found in DNA, this molecule we just saw. DNA you can think of as being, let's say, the master plans for a very complex skyscraper, let's say. But let's suppose something happened to the electrical system in this very complex skyscraper. So you call in an electrical engineer to fix the problem, and they say, "Well, I don't care about all the architectural plans and the design of the hallways and things like this. I just want the electric circuit part of it." So they'd get somebody to Xerox off the parts of the architectural plans that describe the electrical circuits. Well, biology does the same thing. It has a machine called RNA polymerase that goes along DNA and then spits out in certain regions of the DNA another molecule called messenger RNA, and that RNA is like the transcript that tells you about a particular gene itself, not the whole organism, but just about a particular gene. And then that gene itself is then read by another machine called the ribosome that goes along the mRNA, and it spits out proteins. And the proteins are what build you. Now, the messenger RNA is built from those four nucleotide bases, like the ones I just showed you from Watson and Crick. There's one slight difference, but it's not important for this discussion. And then the proteins are built from 20 amino acids, and those 20 amino acids are coded by the nucleotides. So here's a picture then of what happens. I'm going to put on my laser pointer. So here is the machine. It's moving along the messenger RNA like this. Where you see U there, that really means T, if you like. And what it does, it reads it in triplets called codons. And then what it does, it reads it like this. So here you can see it's just read U, U, U. It then knows that that's the instruction to make the amino acid phenylalanine, and then attach that phenylalanine to the protein that is coming out of this ribosome. And that's the process of translation. So the genetic code is the map used by the ribosome to translate the message from mRNA triplets of 4 bases into the 20 amino acids of life. Got that? So it literally is a code book. It says you have an alphabet of 4 letters. I want you to code with that in triplets the code book for 20 amino acids of life. And of course, you've got 4 bases, and you've got 3 spaces in each word. That's the codons. So I've got 4 times 4 times 4, which is 64 possible words that you could make. And that's more than enough to delineate the 20 amino acids of life. So here is the genetic code table. I called it the canonical genetic code because it's basically shared by more or less every living organism, with some minor exceptions. So here is the one we just talked about, U, and then you read this third column here, U, and that gives you phenylalanine. If you had U, and then A, you would get leucine, and so on and so forth. And this stop thing here tells you that that's the end of the message, and then it stops translating. So now I want to tell you about Carl Woese. Carl Woese was my collaborator, and I worked with him for the last 12 years or so of his life. He was one of the greatest evolutionary biologists of the 20th century. And 21st, I suppose. And here he is shown in characteristic pose in his office at the University of Illinois on November the 3rd, 1977. And why? Well, because scientists, led by him, discover a form of life that predates higher organisms. And what he had discovered was a thing called the archaea. So he had discovered that life really comes in 3 domains. Think about this. You're used to the idea of a family tree. So you could say, let's build a family tree of all life on Earth. And so Carl Rose in the 1960s set out to do that. But he didn't want to do it by looking at the the stripes or the number of legs of some creature and things like this. He wanted all of life, and by that point we knew that most of life was microbial. So microbes don't have very much distinguishing characteristics, so he had the idea that we will look at molecular sequences, and he looked at a particular sequence of the ribosome to do his work. So what he discovered was that the life was tripartite. There were 3 domains of life. There's bacteria, that everybody knows. There's eukaryotes, and there's you over there. And then there's this new domain of life that he had discovered. He discovered them. They look kind of like bacteria if you don't look at them too closely, but their evolutionary history is completely different, as you can see here. And that was the reason why he was on the front page of the New York Times, and this was a discovery that has really transformed biology. It was an early form of genomic sequencing, and we're still doing that today. Today we think that this tree is slightly modified, that in fact the eukaryotes, that include us, really emerge not from the root here, but actually emerge from a certain type of archaea species that I'm not going to go into. But more or less, this is the picture. And this root of the tree is called the last universal common ancestor. That organism or community of organisms would have been the ancestor for all life on Earth. And we know that all life on Earth that we know about is related. And this is the family tree of it. Now, what Rose and Fox discovered, I'll show you a picture of them in a minute, was something very surprising. They discovered that the last universal common ancestor, the depth of those branchings, they could already date it and figure out that it was at least somewhere around 3 billion years. And this was very surprising because the Earth is only 4 billion years old, 4.6 billion years. So it's very strange that you could get something like the complexity of modern organisms in such a short period of time after the beginning of the planet. And in fact, there's really 3 big problems in early evolution. So the first one is the fact that there is only a single canonical genetic code. The second one is the one I just said, that the last universal common ancestor is 3.8 billion years. Some people today think it's even earlier. And the Earth is only about 4.6 billion years old. So how did this basic architecture evolve so rapidly and afterwards relatively slowly? And there's a third thing I'm going to show you, which is that the genetic code is close to the theoretical optimum in minimizing errors in gene expression. And the question is why is that? Now, this idea that there is a single canonical genetic code was not surprising to early evolutionary biologists, for example, like Francis Crick, because Francis Crick thought that, well, if we go back to our picture here, that was actually discovered long after Francis Crick was thinking about this, but people assumed that there would be some last universal common ancestor. And Crick had the idea that whatever genetic code the last universal common ancestor had, all the other organisms that descended from it would have to have the same one because they descended from it. And if the code somehow evolved, then you would make the wrong proteins. And if you made the wrong proteins, then the organisms would die, or at least be not optimally fit. And so Francis Crick called the genetic code a frozen accident. An accident because it's just what happened from the organisms from which we were descended, and frozen because it can't evolve, because if it evolved, then you make the wrong proteins and the organisms don't work. Now, what was Rose and Fox's understanding of the solution to the paradox that they already pointed out in their early paper? So here's a picture of George Fox, who's now at the University of Houston. This is Carl Rose doing some of this very, very tedious, somewhat dangerous genome sequencing using what are now very primitive technologies. And they couldn't understand this, and so what they said was, we feel it's more reasonable to assume that the mode, not the temporal evolution, must have changed. In other words, the mode of evolution may have been different in the early eras of life, the first 500 million years or so, than it was now, and the pace of it didn't change. And you may be wondering, well, what do you mean by mode of evolution? What do you mean by that? Well, let's talk about that. So you're used to the idea that you have children and you give your genes to your children, their mother and the father, and then they give them to their offspring and their grandchildren and so on and so forth. So that idea then is called vertical gene transfer. Your genes are transmitted to your offspring, vertically, as it were, down through the ages. But there's a different mode that is known. It's called horizontal gene transfer. So what's happening here is a picture of horizontal gene transfer in bacteria. So here is an example where you have a bacterium and it is infected by a virus called a phage. And what that phage does, I'm not going to go into the details of it, is it goes into the cell, the bacterial cell. It replaces it inserts itself into the molecular machinery of the cell and uses the host to make baby viruses. Then it bursts open the cell, kind of like the alien did in the science fiction movie, and out come 100 viruses that then go and find another cell to colonize. But when they do that, the baby viruses take with them their own viral genome, but because it's a very fast and careless process, they also take with them sometimes some of the genes of the original host, of the original bacteria. And so those genes from that bacterium can then be transmitted to another bacterium, and that's called transduction. So genes can go from one bacterium to another that it's not related to, mediated by a virus, mediated by a phage. There's another thing that they can do, which is conjugation, where little loops of DNA called plasmids can go from one bacterium to another. And lastly, bacteria can sometimes just uptake free DNA that's lying around in the environment, maybe from a burst cell or something like this. And that's called transfectional transformation. So this was discovered, these modes of evolution were discovered in the 40s and 1950s. And the question you might want to ask is, if microbes can do this, okay, that's fine. We can see that they might do this. Probably it's a kind of rare and strange thing, but what happens when they all do it? Well, the answer is very catastrophic. So here's one example. This is the spread of antibiotic resistance genes. So you're probably aware that the most urgent health crisis facing the planet is not HIV, it's not cancer, it's not even COVID. It's the spread of antibiotic resistance genes, genes that enable a bacterium to be immune to antibiotics. And those are spreading rapidly throughout the world, and they are a severe threat to human health because there's some diseases now that are immune to all attempts to cure them by antibiotics. There's some strains of TB, for example, or gonorrhea, that we do not have antibiotics able to stop them. And of course, if this were to go on unchecked, you wouldn't even be able to do medical surgery because you'd get infected and so on. So this spread of antibiotic resistance genes, they're spread by horizontal gene transfer, and that is happening in your guts right now. Okay, so let's now go back to the canonical genetic code, and let's ask ourselves a question about this. What people wanted to understand was what is special about this, and Carl Woese discovered chemically that there is something special about it. He found a way to measure the chemical properties of the amino acids, and he discovered that some of them hate water and some of them like water, and that the amino acids that like water and the ones that don't like water, they are ordered in a particular location in this genetic code table. So it doesn't seem to be at random. And he was able to quantify that with some number, and I've shown some of these numbers over here, basically numbers that start off small up at the top and get bigger as you go down to the lower right. And he did experiments to measure that, and I'm not going to go into the chemical details. Now, I told you that the genetic code was universal, and I said to you that it's nearly optimal in minimizing errors, and I want to tell you why I said that. So we only have one genetic code to play with, but in a computer we can make 20 factorial 10 to the 18 possible codes by just permuting the labels. So you can do that in 10 to the 18 possible ways. And so I can make synthetic codes in a computer just by Monte Carlo simulation. The next thing I can do is I can ask, how good are those codes at minimizing errors? Now, why would you want to do that? Well, you might say, let's think about evolution, survival of the fittest, supposedly. So maybe the best organisms are the ones that if there is a mistake in translation or a mutation, the effect of that is it produces virtually the same kind of protein that it otherwise would have done. So then hopefully that error or that mutation won't cause too much damage. So you replace one amino acid by something chemically similar. So what you can do is you can do a process where you swap amino acids around and you compute the sum of the squares of their polar requirement numbers, the ones I showed you that Carl Lewis had measured. And when you do that, you can make a probability distribution that tells you for all these different genetic codes, how often do I see a particular score, a measure of quality of that synthetic genetic code. Now there's two outcomes of this measurement. One is shown over here. So I'm plotting the probability distribution of a property of the code. In this case, it is the score that tells you how good the code is at minimizing error. And there's many different ways you can construct this measure, but the answer is the same for whichever one you do. In this particular measure, the smaller number is the better one at minimizing error. So you might get a distribution that looks like the one I'm showing you over here. And if the actual genetic code has a value that's, say, in the middle here, like I've drawn with this line, then you would say, well, the current code is just one that is pretty much like what you would get if you just generated codes at random, and then you would see that they would most likely be somewhere in the middle of this distribution, and there's the canonical genetic code. On the other hand, if you got this, so here's your distribution of random scores, but over here, this is what the actual genetic code is, then you'd say, well, that's really weird. It's not an accident. It's not random. It must have evolved. So it's not a frozen accident after all. So what happens when you do this experiment? So here is one of the early measurements of this. This was done by Freeland and Hurst in 1998. And what they discovered was here's our random genetic codes that you can do. And down here in the tail is the actual canonical genetic code. The title of their paper was "The Genetic Code is 1 in a Million." In fact, we did this calculation using different methods and so on. It doesn't really matter. It's more like 1 in 100 million. It's a very, very rare thing. And it tells you that it could not have evolved by chance in its current form. It must have evolved. But if it evolved, how could it evolve? Because you would still be subject to Francis Crick's frozen accident paradox. If the code evolves, then you're going to make nonsense proteins, and then the organisms will die. And that is the problem that we solved in this paper in 2006. And I'm going to tell you what we did in that paper. So I first of all want to give you the physical insight about how it is possible for a code to evolve. So let's have a look at the problem. So I'm going to do this by an analogy. So I'm going to talk about who are the experts on codes. Well, those of you above a certain age will recognize the people shown in this picture here. These are secret agents from like 1960s and 1970s TV shows and movies. And of course, secret agents, they're experts on codes, and they communicate using some secret agent code book. And so I want to do a thought experiment when we ask how could secret agents communicate using a code book with this particular member of the secret agent fraternity. And we're going to go through a series of experiments, thought experiments, to see how do codes work and how they can evolve. So let's suppose that we have the following situation. So here is our secret agent who wants to transmit this message to the person on the right. So the person on the right gets the message, they get out their secret agent codebook, and they decode the message, and the message says the following set of amino acids, and that's all well and good. Okay, that's how it should work. That's how you probably know codebooks evolve. The thing is this agent, as you probably know, has lots of obnoxious characteristics, including the fact that he's very clumsy and careless and so on. So let's imagine that his code book has lost half the pages of it. So even though the real code book would work and would give you only a single message, when um, when you've lost half the code book, the message doesn't make any sense. So he takes this message and he reads it and it says to him E equals mc squared. What the heck does that mean? Okay, so the code, then the message that you get will change, and the message gets garbled if you've somehow evolved the code book. And the evolving of the code book is shown here by tearing out half the pages of it. Okay, so the message gets garbled and Francis Crick was right. So that message doesn't make any sense to him, might make sense to the physicists in the audience, but that wasn't the actual intent of the message. But suppose you had a probabilistic codebook. So your codebook is not one which just gives you one single message, but gives you a whole set of messages. And then you have to look at those messages and from the context, deduce which one is the most likely one. So in this particular case here, the receiving secret agent has done some probabilistic analysis and has come to the conclusion that it's not clear what the message was, but with high likelihood it referred to these three amino acids. And so what you've learned from this is that if you have a probabilistic code book, then you can still deduce messages even if the messages are actually evolving, because the correct message will be interpreted, at least it will be visible amongst the ensemble of probable messages. Now this then tells you that you could then have an algorithm for having codes which evolve. And the idea was already figured out in some form or other by Carl Woese in the 1960s, and he called it statistical proteins. Think of it like this. When you had early life would have been an organism that was, not like today's organisms like us, which are very, very complex and have brains and things like this. Early life was round blobby things, and they just sort of floated around in some soup or whatever, and they had very primitive cellular machinery. They were kind of like very early automobiles. Early automobiles were, couldn't go very fast, and they would always fall apart and so on. And let's suppose, I were to drive around to your house in an early automobile in the late 1800s, and let's suppose the wheel came off of my car, you would say, well, don't worry, I've got a spare bicycle in the garage. Why don't you just take the wheel off that and stick it onto your car? And you could do that. But you certainly wouldn't be able to do that today if you did that with a Toyota Camry or something like this. You need to have very specialized equipment. So basically, what you've learned from this little anecdote is that very complex machines need very complex codes, and very simple machines can get by with very simplistic codes, even probabilistic ones. And so you have the idea then that life, as it evolved in complexity, would also have evolved the machinery, the underlying software platform of life, and that would have co-evolved along with the complexity of the organism. So how do you then get selection of the code in this way? So let's talk about competition between innovation sharing protocols. So on the left here, I'm showing you an early picture of Bill Gates with an early form of the Microsoft computer operating system. And here is Steve Jobs with one of the early Apple computers. And these two operating systems competed until very recently. They still compete, in fact. And the fact of the matter is that I don't know what the numbers are now, but certainly a few years ago, it was like 95% of the world's computers ran the Windows operating system, and only a small percentage ran the Apple operating system. And you might say, well, why is that? And you might say, well, it's because the Windows operating system is much better. We just have a problem. My Windows computer appears to have crashed. I have a blue screen of death. Well, maybe it's not that the Windows computer is better, the Windows operating system is better after all. Maybe there's something else going on. What could that something else be? Well, that something else that could be is that The thing about the Windows computers, the Windows operating systems, was that when you were a software developer in the 80s and 90s and early 2000s and so on, if you wanted to code and sell your code, the most number of users were people who used Windows computers and you know, IBM PC architectures. And so so therefore, if you wanted to have a software package that, I don't know, did your taxes, for example, you could easily get that if you had a Windows computer. But the programming companies, the software companies, wouldn't necessarily make that for the Apple computers, because they were, you know, such a small fraction of the population that there's no point to do that. So what you learn from this, well, actually, this lesson, by the way, is a correct lesson. And when Steve Jobs and Apple developed the first smartphones that we use today, the iPhone, in the first decade of the 21st century, one of the first things that they did was start up the App Store. And it was the App Store that gave the iPhone its competitive advantage. And it was many years before the Android operating system was able to catch up. So what you learn from this is that the community with access to the broadest range of innovations has an evolutionary advantage. And that evolutionary advantage is due to the platform, the software, not what you can do on it. And in the case of life, that is an innovation sharing protocol. That's what essentially software is about. And so that's the genetic code. So we looked at this by studying simulations of genetic code evolution. And I'm not going to show you how we did it. Here's a bit of our paper. There's lots of math and theory in it. What we basically did was we followed what I showed you before, this first digital life simulation. We made an asexual population of evolving, reproducing digital organisms. We made them have digital proteins. Better proteins give more offspring. The proteins are obtained by translating the genome with code, with errors. And the question we're trying to understand is, will evolution cause these digital organisms to evolve a unique and optimally translating genetic code? So here's our results. On the vertical axis here is the quality of the genetic code, how good it is in minimizing errors. This is a function of time. And over here in red is what happens when you just do vertical evolution, the same evolution that gives rise to a tree of life. And you can see that as I had told you, that eventually evolution stops and you get a frozen accident. And if you do it again from a different initial condition, you go down a different trajectory and you come over here. You do it again, you get a different trajectory, you go along here. And so this is what happens. This is exactly what Francis Crick predicted, the frozen accident hypothesis. And if you look at what you get with this, you see that the result is indistinguishable from what you would get from a random code. But if you switch on horizontal gene transfer, then you can see that what happens is that the code eventually evolves to one that is very, very, very good at minimizing errors. And in fact, it is unique as well. So the horizontal gene transfer leads to optimality, and the horizontal gene transfer leads to universality. Okay, so what we've discovered then is that vertical evolution is not fast enough to produce the observed unique and optimal genetic code. Horizontal evolution is fast enough to produce unique optimal genetic codes, and therefore the ability to form complex cells. So you have a phase diagram of life that looks something like this. Here is time going up the screen here. This is a sort of sketch of this early phase of life where you have horizontal gene transfer of the machinery of the cell, the core machinery of the cell, the translational machinery. And because everything can send bits of genes to organisms which are not related to it, you don't get a family tree. Instead, you get a network. Then something happens, the mother of all phase transitions, and you get a transition to a tree dominated state, which then evolves slowly. The evolution is fast here because you have network effects, just like you see today in social media. Social media, rumors spread very rapidly through the network effect. And that's the same thing is true for genes as well. And so the phase diagram of life looks something like this. You have a last universal common ancestor giving rise to the modern trees of life. But early on, there was a communal state of competing codes, and those codes then competed, and the ones that had the greatest access to a pool of innovations were the ones that won, and then you ended up with a communal state with explosive growth in innovation sharing, giving rise to the modern form of life that we see. And this explosive growth in innovation sharing is exactly what happened to the internet in the middle 1990s, once the hypertext transfer protocol was developed by scientists at CERN, for example. So I'm not going to go through. Well, I'm not going to go through this in detail, but I will tell you a little bit another very quick anecdote before I finish. So I want to tell you about homochirality. So homochirality is the idea that if I look at the biological amino acids of life, they're all left-handed, and the biological sugars are right-handed. This is a 100% effect. It's not just like, well, there's slightly more of left-handed ones than right-handed ones. And using theoretical physics, we were able to show that this symmetry breaking between left and right-handed could work by studying the properties of order-catalytic self-replicating molecules that will automatically lead to a homochiral state. So what this tells you is that life is out of equilibrium, and that was important in our work. And it shows that the emergence of homochirality is inevitable, and so it can be a universal property of life, even a biosignature. Now, why do we care about biosignatures? Well, this rover will be able, actually may even be on Mars by now, will be able to test for homochiral organic molecules on Mars. It's been instrumented to look for life, and homochirality, now that we theoretically understand that it could be an outcome, a universal outcome of life, not just life on Earth, one can look for that. Here is another wonderful astrobiology discovery that was made in the last 10, 15 years or so by the Cassini satellite. What you're looking at here is the ice crust of a moon of Saturn called Enceladus. And what we already know by looking very carefully at the way this planet orbits, the Enceladus orbits the planet Saturn, what we already know is that it is inside, on the top it is an ice crust, but underneath that ice crust, it has a global ocean of liquid water. And we know that because it wobbles in a way, just like an egg would wobble if you spun it and then put your hands on it to stop it spinning. If it was in the liquid phase, the water or the stuff inside the egg would still be spinning even if you stopped the shell, and so the egg would then start to spin again. So having something inside that is liquid will affect the way that things rotate, and that can be predicted using physics, and that was observed by the scientists monitoring the data taken by the Cassini satellite. So we know that there are global oceans of liquid water under Enceladus and also Europa and perhaps Pluto as well. They're not the only ones. And we know why, because they're close enough to Saturn that the gravitational tidal forces can actually cause the rocks at the core of the moon to melt. But there's more, because actually there's cracks in the ice, and those cracks in the ice have fountains of water squirting up into space, and we can see them. Here's the picture here. And not only can we see them, but the scientists running Cassini sent it through those fountains. It wasn't instrumented to look for living matter, but it could establish something about the water chemistry there. And the water chemistry there was non-equilibrium, and it resembled what we see in alkaline hydrothermal vents on Earth in the deep ocean. And those are the environments where many of us think that life could have evolved. And so the question we'd like to know is, are the molecules, the organic molecules in these fountains, homochiral? So let's conclude. What can theoretical physics tell us about the evolution of early life? Well, simple models can predict real biological phenomena. Yes, if you ask the right questions in the right way. The take-home messages are that theoretical physics starts with a simple guess, then you calculate the consequences of that guess, and you compare with experiments or predict a new experiment for someone to do, and those experiments suggest new guesses and so on and so forth. That's the scientific method. Simple models, but not too simple, can describe very complicated phenomena. They can understand, say, superfluid flow in 2 dimensions and things like this, and it can understand some of the generic aspects of the emergence of life that's to do with how information is transferred, which is an essential part of what living systems do. So here is Arnold Sommerfeld, a very famous theoretical physicist from the early part of the 20th century. He said, if you want to be a physicist, you must do 3 things. First, study mathematics. Second, study more mathematics. And third, do the same. Okay, but I would say it's not quite like that. If you want to be a biologist, you must do 3 things. First, study mathematics. Second, study more mathematics. And third, do the same. Well, not all biologists should do this for sure. But the point is that biology is a field that can be mathematized and can be understood by the same sorts of techniques that have been so successful in theoretical physics for some questions, but not all of them. And I'd like to end with this quote, which I find very inspiring, by Stanislav Ulam. He says, "Ask not what physics can do for biology, ask what biology can do for physics." He's paraphrasing John F. Kennedy there in his very inspirational speech from 1962 or 1963. And what this means is, for physicists like me, I study biology not just because I'm interested in biology per se, but because the study of biology expands the scope of physics, and I think that's a very exciting thing. And so I want to leave you with that message. Here is some reading material if you're interested in the perspectives that I've given you in this talk, and I'll be happy to conclude and take any questions.
Thank you so much, Nigel. That was a beautiful talk, really interesting concepts and great presentation. There are some questions. So if you have questions, go ahead, type them in. There's a question from Shiva Agarwal, who asks, does evolution have any preference for horizontal or vertical gene transfer?
No, they both go on all the time. They're both things that can happen. Anything that can happen is not forbidden, will happen. In the early form of life, it was just as easy to transfer core components of the cellular machinery, the translational components, as it is today to transfer horizontally antibiotic resistance genes, for example. But today, now the machinery has got so complicated that it is very difficult for core cellular machinery to be horizontally transferred. If you try to send in some new gene, the likelihood is that either you've already got it, or the machinery is so complex that you just simply can't incorporate it into it. And so horizontal gene transfer for very core cellular function rapidly gets frozen in. But the non-essential genes, such as antibiotic resistance and other genes, those are free to transfer, and they are transferring all the time right now. So I hope that answers the question.
Yep. So next question is from Ten Fu, who asks, what can biology do for physics?
What can biology do for physics? Well, I would say that's not the purpose of biology, but it is the purpose of physicists who are interested in this to not feel that physics is only the study of the inanimate universe. The physics is the study of everything that happens within the universe, of natural phenomena, of which life is one of those. So those physicists who are interested in understanding the phenomenon of life, which is, I think, is the most, as I said in the introduction, the most remarkable thing that you would not have expected. I mean, how would you have known that matter self-organizes into hierarchical structures that have lots of feedback loops between them, that are capable of evolution, that are even capable of advanced functions like consciousness and intelligence and things like this? We don't have any basis for understanding that, and it's even quite still difficult to understand what it is that makes living systems become more and more complex. We know that they do. We have unequivocal proof that that is what has happened in the past. And we have some simple models such as ones that I've made and others, that show you how you can, in principle, get these open-ended growth of complexity. But the problem is far from understood. And so, why we don't have, why the world isn't just knee-deep in self-replicating RNA slime. Why do you have elephants and humans and things like that? So I think the phenomena in biology are nature already gave us those phenomena. It's our job as physicists to study them. For biologists, what can they do? Well, I think the thing that they can do is that when a physicist calls and says, I'm interested in learning about biology, the biologist should talk to them. And they do. I've had wonderful collaborations with biologists and still do. And I think that, breaking down these silos between the disciplines is very important. Probably the things that I do as a physicist are not going to be the things that cure cancer, for example. That would be a biomedical researcher, but then most of what biologists do won't cure cancer either. So there's a whole continuum, and physicists can have their parts to play in all of those. Let me give you another example. The publicly funded Human Genome Project was funded by, or was led by a person whose PhD degree was in pure mathematics. Okay, not a biologist. Okay, and many people involved in it were physicists, computer scientists, mathematicians, and so on. Some of these problems in biology really involve very complicated mathematics. For example, how do you assemble genomes and things like this? So I think that there's beautiful mathematical problems in biology that are just waiting for people like me and others to sort of turn our attention to them, and we can't solve all of them. So some of them we can make a contribution to.
Okay, great. So there are actually a lot of questions now. So let me just take two more questions, and then people can hang around afterwards and ask questions privately. So there's a question from Stephen Vigdor, who asks, is homochirality the sole selection criterion for life on Earth, choosing 20 or 22 amino acids out of a much larger class of amino acids?
Is homochirality the sole selection criteria? No, I wouldn't think so. I mean, we don't. The homochirality is a funny thing, because once you have some homochirality, then you can think of reasons why having homochirality is a good thing biochemically. For example, it enables certain types of packing of molecules and folding and things like this. So you can sort of say post hoc, well, if you had homochirality, it would be a good thing. So there's an evolutionary selection pressure for that. But it turns out that even without that selection pressure, you can still have the evolution of homochirality. So I wouldn't say it's the main selection criterion, no.
Okay. One last question from Bernd Mueller. Asks, is there a similar explanation for the fact why the slow evolution after the last universal, so the Luca,
last universal common ancestor,
yeah, common ancestor, suddenly transitioned into the explosive evolution of higher life forms in the Cambrian 3 billion years later?
I don't think so. Thanks for the question. So one of the things that I've neglected in talking about here is ecology. And the way ecology interacts with evolution is very complicated, and I've given lectures. Some of them are available on YouTube, where you can learn about that. But basically, when you have ecological niches after, say, a mass extinction event or something like that, there are a lot of available niches for organisms to colonize and take over. And so a lot of the I'm not a huge expert on these things, but a lot of these rapid growths of diversity follow extinction events.
Okay, well, thanks so much, Nigel. This was fascinating, and thank you for answering the questions in a beautiful presentation. This will end the public part of the session, so thank you everybody for coming. For those of you who want to stick around, please do so. I will let you talk with Professor Goldenfeld. So let's thank once again Professor Goldenfeld, and have a great weekend.